Join medicinal chemist and In the Pipeline blogger Derek Lowe as he reveals the 7 steps and essential questions in drug discovery.
I'm going to give an overview of drug discovery. So this takes us with everything all the way up to the clinic. First one, disease pathology. This is a high-level strategic question. What therapeutic area do we work in? What disease are we targeting? The next step is target ID. That is more specific. This is when we start saying, what exact process are we going to target in this disease area? Now, once you've done that, your next step is assay development and screen. The big question here is, what kind of assay are you going to run? Do you have a specific molecular target in mind, or are you going to run phenotypic, looking for the desired effect in some organism model? That takes us to the next step. Step 4 is hit to lead. You've run your screen, and now you have the hits. The idea is to triage these hits into the most interesting, things that are possibly interesting, and things which really just have to be pushed aside. Now that you've narrowed down, you then go on to lead optimization. Traditionally, lead op is classic medicinal chemistry. This is really the part where everyone has to start improving the classic properties: potency, selectivity, toxic side effects, and pharmacokinetics. A few months would be the shortest lead op that you can imagine, and a few years is not unheard of either. But once lead op has narrowed things down to just 1, 2, or 3 clinical candidates, then you move on to preclinical development. You've got the classic scale-up questions. Can this compound be scaled up at all? What shape is the route in? Can the reagents be sourced? Is the yield and the purity profile reproducible? You also have tox to worry about. The standard is 2-week tox in at least 2 species, usually 1 small one and 1 large one. And finally, that takes us into the clinic, which is, of course, our goal. The clinic is where the real money gets spent in this business. Everything that happened preclinical, those are round-off errors compared to what you spend in the clinic. That doesn't mean that earlier stuff isn't important. That means it's critical. That is the leverage point for the whole process. If you don't get those things right, you run a risk of wasting vast amounts of money and vast amounts of time in the clinic. So all of these details before then have to be done well to the best of everyone's ability.
Join us live every Thursday at 2 p.m. Eastern for the best and brightest minds in chemistry.